The University of Michigan Division of Gastroenterology and the Inflammatory Bowel Disease Program host the IBD Visiting Professor Lecture Series in Ann Arbor, Michigan. This series presents the latest in clinical research and basic science from IBD experts from around the world.
Thank you so much, Peter. Thank you so much for inviting me. It's been a terrific day yesterday, and I'm looking forward to the rest of today. I come from the other UM, I suppose, the much younger UM, actually. As I was getting walked through your corridors yesterday, I think your medical school is literally 100 years older than ours. So it's a real honor for me to be here. And I didn't appreciate initially that I would be talking to, a mixed audience in that this is your clinical grand rounds. Obviously, I wear both hats because I see patients, but I also do basic science. So I'm hopeful that all of you will get something out of that. And so this is what Tim will look at the rest of the day, because I'm sure all of his colonoscopies are well prepped and took all of their go lightly, because I suspect people in the Midwest are far more adherent to what you tell them to do. This is more like the colonoscopies that I get to do. And that Ellen and Peter get to do, which is in this case happens to be ulcerative colitis. But basically, one of the things that drives the research in our laboratory is to understand how it is really that we all normally coexist happily with a very high density of bacteria, and why is it an aberration then for patients to develop inflammatory bowel disease? Is it a distinct microbe? I had an interesting conversation with Vince Young. Is there a distinct microbe? Or is it an aberrant response to the normal flora that's there? And one of the things that has emerged, and even is kind of a brand new field since the time I was in medical school, is the whole topic of innate immunity. And basically, we have hard-wired receptors that are meant to recognize pathogens or pathogen-associated molecular patterns immediately. And once you come in contact, this is a macrophage with tuberculosis, mostly because it was a really pretty picture that I could download. And these receptors then activate a whole cascade of events resulting in cytokine and chemokine production to recruit inflammatory cells that are then responsible for trying to eradicate that pathogen. In addition to eradicating that pathogen, they're absolutely essential for priming an adaptive immune response. Without innate immunity, you can't prime an adaptive immune response. And in fact, although people thought of these two processes as being on separate cells and completely disparate processes, we and others have even described that T cells actually can express toll receptors, so that it's not so neat and tidy that these are the cells that make TLRs and these are the cells that make the B cell receptor and T cell receptor. All right, and so we got interested in this field when toll receptors started to be discovered. So these toll-like receptors, it's a funky name because they were, if you're interested in the history of it, I know Linda Samuelson's husband is interested in the history of medicine, but at least the history of tolls is that tolls actually is the German, like some German exclamative for like holy cow. And they discovered a Drosophila that was completely inverted in its structure, and so they exclaimed toll. And then this toll ultimately was discovered to be very important for innate immune defenses in Drosophila, and you can see how this has been conserved through higher and higher orders of species, including man. And so there are at least 12 tolls. The first one that was described is TLR4. Toll-like receptor 4 is absolutely essential for the recognition of endotoxin. And these toll-like receptors are variably expressed by antigen-presenting cells like macrophages and dendritic cells, but we and others, of course, have described they're present in intestinal epithelial cells and more recently even in T cells. And the list is too long to put on one slide. Most of all these different toll-like receptors recognize different pathogen-associated molecular patterns. So even though TLR4 is required for LPS, you can see that things like gram-positive bacteria with a lot of peptidoglycan are recognized by TLR2, sometimes in combinations with TLRs 1 and 6. And most of them use an adapter molecule called MYD88 to then bind to other kinases that eventually culminate in the activation of NF-kappa B and a lot of the cytokines we've come to know and love that are pro-inflammatory. TLR4 is unique in another way, which is that it also can activate a parallel pathway using a different adapter molecule called TRIF that results in interferon alpha secretion. Interferon beta secretion, excuse me, and that pathway is also used exclusively by TLR3. So you can see that we're still this, I could have chosen to show one of those seizure-provoking slides of the network of things that are activated by TLRs, but distilled, this is how TLRs function to have the correct inflammatory response. And this I show kind of out of a sensitivity to the notion that, oh, well, it ends up being NF-kappa B anyway, so we know a lot about NF-kappa B, so who cares about that? But in fact, as you might imagine, there are lots of inputs. NF-kappa B is a central transcription factor that really integrates a lot of disparate signals coming into the cell, only one of which are the TLR signals, things like TNF and IL-1, and lots of things, the list was as endless, can activate NF-kappa B. So there's that Venn diagram, and then TLR4 is not only capable of activating NF-kappa B, it can activate other things. So this is why we felt it was important, at least to focus our work on TLR4, and this is sort of the lead up to tell you what we've been doing. So it should be self-evident that if we are coexisting with such a high density of bacteria, there must be a reason for this, and it's only the exception when this comes back to be detrimental. But what I would say is that what I think has happened in the gut with respect to coexisting with bacteria is that, in fact, we've usurped the presence of bacteria for good, normal, healthy functions in the epithelium. So why do we have bacteria? Well, this is not the reason why we have bacteria, but the fact that we do have bacteria, bacterial recognition, at least in part through TLRs, is really essential for epithelial repair and restitution in the gut. That is our work and the work of others, that in fact having, when you have a hole in your membrane, it really helps you to know that you have bacteria that are coming into the wrong location, and that signals a whole series of events to try to seal up that hole. In addition, bacteria are involved in extracting more nutrients from the foods we eat. And in the gut, again, rather than having a pro-inflammatory response to bacteria that have found themselves in the wrong location, by and large, these bacteria are phagocytosed and cleaned up neatly and tidily so that they don't trigger a big sort of detrimental response to the host. This is a slide stolen from Gabriel Nunez, not yesterday, but months ago. I could use it because I know that he couldn't make it here this morning. But the idea being that, I guess some of you know why he's not here this morning, that in fact we need an inflammatory response so that we can get rid of pathogens. But if that inflammatory response goes haywire, then of course it could be damaging to the host. A good example of this would probably be Plasmodium falciparum. We don't die of the actual malaria, we die of the immune response to the malaria, especially in the brain. I've oversimplified a lot, and probably I'm not even correct, but you know, Crohn's disease, at least all of the genetics are telling us that this is defective innate immunity. And perhaps to some extent, ulcerative colitis, maybe not as a primary defect, but eventually results in exuberant innate immunity, which again is detrimental to the host. So perhaps not surprisingly, many of the genes that have been uncovered, both using candidate gene approaches as well as using genome-wide association studies, which is the latest way in which we've made progress in the genetics of IBD, implicate innate immune pathways. And I think that makes sense given the fact that you would expect that if you had a perturbation in innate immunity, it would disproportionately affect the surface that is most contaminated with bacteria. We have focused on TLR4 for a number of different reasons, including the fact that it was the first one, and from the very beginning when I heard about TLR4, it occurred to me that, gosh, how is that regulated in the intestine, since TLR4 recognizes LPS, and we've got tons of LPS, so there must be something special about how TLR4 is regulated in the gut. But there is genetic evidence that it has some role in Crohn's disease, and polymorphisms in TLR4 are more common in Crohn's disease patients, but not by a clinically meaningful amount. In fact, none of the genes that we're dealing with, except for perhaps NOD2, unless you use it in some kind of a grouping or for phenotyping, are predictive of who's going to develop IBD.